Okay, good day, viewers. Uh, Thanks for following the channel. If you're not done yet, you can actually do that. So today we want to talk about biology, and I want to talk about a specific part of biology, a very important part actually, organization, organization, organization of life. How is life being organized? And of course, when you talk about organization of life, what do we mean? We mean life in stages because life is lived in stages. When scientists study life or an organism, let me not use life, let me use an organism. When scientists study an organism, they are studying that organism in different stages. What are those stages that they study organism? They start from the simplest level, which of course is a cell. Then they now move to the next complex or the immediate complex level, which is the tissue. Before they move from the tissue to the organs, from the organs to the systems. After all of this, that's when you can now say you studied the what? The organisms or the organism for singular. Now, what does this mean? This means that see, cell is the smallest structural and functional unit of life that can be studied. Tissue is the immediate brother or immediate senior brother to cell. What does this mean? This means that a tissue is made up of components of cells. So when cells come together, they form what we call a tissue. So this tissue now comes together to form what we call what? Organs. The organ comes together to form what we call system, and the system comes together to form what we call what? Organisms. Now, each of these levels have organisms that lives in them. For instance, we have bacteria. Bacteria lives as a cell, a single cell for that matter. Bacteria live as a single cell. Some protists live as a colony of cells. That is a cell or many cells will come together to form a colony. That does not mean that that organism is multicellular. It means that the organism, maybe for instance, uh, a group of army men comes together to form a barrack, isn't it? Right? Does that mean that all those army men are one? No. It just means that those army men, they are living as if they are one, but they are not one. So that's how this thing, colony works. Some organism lives as colony in the kingdom protista, most especially. We also have, of course, the tissue. Some organisms live in tissue like the hydra. We have the organ like the bulb. You check your onions bulb. The onions bulb is in the organ level. Okay. You also have the system and the of course the organism. Like me and you, we live in the organism. We are a full organism. We have system. We have tissues. We have organs. We have cells, and so on and so forth. So in this particular video, we will delve deep into cell, and we will now make another video for tissue, another one for organs and system altogether. All right, we don't want to rush things. Okay, so let us talk about cell, for instance. Let's talk about cell. I said cell is what? The structural one, structural, functional functional unit of life. You see those three parts that I mentioned: the structural one, the functional two, unit of life. So if anything is happening in your system and you want to trace it, the best thing to study is the cell, because every single function of an organism starts from the cell. As you can see in the definition, every single structure of an organism starts from where? The cell. Now, let me blow your mind. You see plants. The reason why plant is, yes, mommy, I know, 1, 2, 3, 4, 5. Let me blow your mind, for instance. The reason why plants is structural the way it is because the cell wall is not made up of the same material that we have in plants. The cell wall, animal. The cell wall of plant is made up of cellulose, a very strong material. That's why you see plants are static. They don't move about the way we and animals move about. All right. Now that we've established that fact, we need to understand the types of cells. There are two main types in every organism. Two main types. We have either the prokaryotic or we have what we call the eukaryotic. Two main types of cell. You see that a cell can be prokaryotic or it can be what? Eukaryotic. What does that mean? Pro means false nucleus. Anything, any way you see prokaryotic means false nucleus. This means that the nucleus is scattered in the cytoplasm. Now, in every organism, there's what we call the nucleus, which is the central system or the most functioning system in the cell. If that system is covered with a nuclear membrane, that is in what? Eukaryotic organism. But in prokaryotic organism like the bacteria, that nucleus is not covered in what? With a nuclear membrane. What does that mean? That means that that organism has a scattered nucleus, right? So if the organism has a scattered nucleus, it is classified under what? Prokaryotic what? Organism or prokaryotic cells. Well, those organism that contains a cell that is defined with a well-defined nucleus, those kind of cell is called eukaryotic cells. So know these things because it's very important. Now, let us give a practical example of what we call about prokaryotic cell. Let's say, for instance, we have a bacteria. Let's use a rod-shaped bacteria, which is bacilli. So we have a bacteria. Of course, we will first of all draw the line twice. This line, drawing it twice here, simply means that we are acknowledging the word cell wall because the bacteria has a cell wall, and the cell wall, like I said before, is made up of what? Peptidoglycan. That's what differentiates bacteria from other organisms. The cell wall of bacteria is made up of peptidoglycan layer, while the cell wall of plant is made up of cellulose. The cell wall of fungi is made up of what? Chitin. You need to know all these things. Now, we now have what we call the flagella. Of course, this is the cell wall. We now have the flagella. What's the main function of the flagella? Used for movement, just like your legs. But in this case, the flagella, it has a whipping movement. So it whips. As it whips, the organism propels. So we also have a very important, and of course, the cell was made up of peptidoglycan, please. Peptidoglycan layers. And of course, we have the lysosomes or the perisomes. The lysosomes. These lysosomes, what is the main function of the lysosomes? The lysosomes is used for breaking down dead matter, or we call it detoxification. Bacteria produces a lot of toxic substances. Because of that, they need the lysosomes to break down those toxic substances so that it will not affect the cell proper. Now, we also have the little bit, let me put little dots. This little dot here represent the what? Represent the ribosomes. Ribosomes. These ribosomes are used for protein synthesis or breakdown of protein-related materials. And of course, you also have the mitochondria. Let's make a elongated curve, something like this. Something like this. These elongated ones are called the the mitochondria. And what is the main function of the mitochondria? It's used for what? For powering the cell. It's for powering the cell. When a particular cell needs energy, it gets its energy from what? The mitochondria. Okay. Now we now go to the nucleus. The nucleus, of course, you have the nucleus, you have substances around it. These substances are around it. All right. Now you can now notice that what I was saying before. You check, there is no nuclear membrane here. There is the nucleus normal. The nucleus will be there. This one. We will also have the chromatin. But if you check very well, you notice that there is no what? There is no nuclear membrane. If there is a nuclear membrane, I would indicate it by drawing a circular, you know, part around the nucleus. We also have the contractile vacuole. We have the contractile vacuole all the way around, scattered around it. Contractile vacuole. We have the contractile vacuole. Now, what's the main function of the contractile vacuole? It is used for what? Osmoregulation. For instance, if this organism produces waste product, how can this organism excrete this waste product? Through the what? Contractile vacuole. And we also have the food vacuole. Let's just use any zero here, any circle rather. We have the food vacuole. Now, this is a typical structure of what? A bacteria. Now, so you now come to realize that you've noticed that there is no, the main reason for drawing this is for you to see that there is no nuclear membrane. There's no nuclear membrane. Okay, let us draw the typical eukaryotic cell. A typical eukaryotic. When I say an eukaryotic cell, I mean a cell that has a what? A cell that has a nuclear membrane and other parts. For instance, the Golgi apparatus is not here. The endoplasmic reticulum will not be here in the prokaryotic. But in the eukaryotic cell, you see all those things. You see all those things. So let us draw this. Normally, we draw something like this. And we now try to double it. Let's see if we can double it perfectly. I'll double it. Okay, let us try. And you know, in an eukaryotic cell, most times it's not always cell wall, it's always cell membrane. Now, if it is a cell wall, for instance, in fungi, it is still a cell wall in fungi. In plant, it is still a cell wall. That's the cell wall is not made up of the same material that we used during the bacteria. Because in the bacteria, we said it is made up of what? Peptidoglycan layer. But in this other aspect, it is made up of, in the cell wall of plant, is made up of what we call cellulose. Alright, it's not made up of peptidoglycan layer, it's made up of cellulose. And of course, this cellulose is what makes up the cell wall of plants. Well, for fungi, it's chitin. Chitin is spelled Chitin. That's chitin. Okay, then we now come to understand something. We say that in an eukaryotic cell, the nucleus is what? Is centered at the middle. With, of course, we have the DNA, we have the chromatin, this will be the cellulose nucleus. We have materials around it of different shapes and sizes. You don't really have to make it like this. You can make it your own shape, your own size, different shapes and different sizes. right? Okay, so we now have the granules. We call it the granules. All right, so this is the nucleus. Let me pick a point. The nucleus. Let me put a place to label on this. Somewhere down here. Nucleus. Nucleus. We have this to be the chromatin, and then the chromatin contains the what? The DNA or the chromosome. Chromatin, which of course that's where we have our DNA and our RNA. Of course, we also understand that we have here to be the cell wall or the cell membrane, depending on the organism. It can either be cell wall in plants or cell membrane in what? In animals. Okay, we now have the smooth endoplasmic reticulum, the smooth endoplasmic reticulum, the smooth endoplasmic reticulum. We have the smooth and we have the rough. What's the function of this? It is used for transport of materials in and out of the cell. It's for transport of what? Materials in and out of the what? Out of the cell. So we have the smooth endoplasmic reticulum. And we have another one here, but that's actually the rough. We have it as the rough. We have it as the rough endoplasmic reticulum. Now, this rough endoplasmic reticulum, what makes it different from the smooth is that it contains or ribosomes are attached to it. So any reticulum that has ribosomes attached to it is called what? It's called a rough endoplasmic reticulum. When we have ribosomes attached to it, it's called a rough what? Endoplasmic reticulum. So this is a rough endoplasmic what? Reticulum. And of course, it is also used for storage of protein, and for protein synthesis, it's also used for transport of protein from one part of the cell to another. Of course, we also have the mitochondria used for what? Energy production, the powerhouse of the cell. So this was used for energy what? Energy production. Sorry, the mitochondria. The mitochondria. And of course, we also have the lysosomes. The lysosomes used for detoxification of materials that are toxic to the cell. I listed the ribosomes. Okay. We also have the ribosomes. I've not listed that. The ribosomes, they are like small dots all over the cell. The ribosomes. The ribosomes. We have this to be what? The ribosomes. Then we have the cytoskeleton. Of course, we have the cytoskeleton, other small organelles. Just use a circle to demonstrate this. Just pick one circle. We have this as the cytoskeleton. Then we have the vacuole. The vacuole in animal cell is not big, but since it's a general cell, we'll just make it medium size. The vacuole in plant cell is very large. Now, why is it large? It is used for storing pap. It's also used for structure. It gives structure to the cell in plant cell because plant cell is very structural, very structural vacuole. Okay, so this is a typical eukaryotic. Let me name it here. There's no place to name it. This is a typical eukaryotic what? Cell. And these are things you need to know offhand. You don't actually have to draw it exactly this way. Okay, I've missed one thing. The Golgi apparatus. Very important. Or the Golgi body. Let me create space here. The Golgi apparatus. Golgi apparatus. Golgi the Golgi apparatus. What's it used for? It's used for transportation of materials in and out of the cell. Okay, so this is just a sketch of what